Today, I will be talking about a project I did as a part of my PhD dissertation at the University of Michigan with Dr. Gina Baucom. More specifically, I will talk about the inter-chromosomal linkage disequilibrium and link fitness cost loci associated with selection for herbicide resistance. So, the first thing we should ask is why is it important to study herbicide resistance in depth? And what are the impacts of the use of herbicide on the plant ecology and evolution? So, the use of herbicide can have various impacts on the community, from leading to weed shifts, to which is the change in the composition or the relative frequencies of weed in the community, to leading to resistance evolution in particular species. Herbicides can also have immediate effects on the ecological interactions like changes in flowering time, palatability to herbivores, and susceptibility to diseases. Or can have long-term ecological changes from resistance evolution and weed shifts. Here, I'm going to focus on this resistance evolution and the genetics underlying this process. Resistance evolution has rapidly increased in the past few decades, with over 245 different species that have developed resistance to a number of different herbicides. This resistance evolution occurred broadly via two different mechanisms of resistance, which I will expand on now. So, in the first scenario, we have the wild type, which is a susceptible case, meaning no resistance. So here we have the herbicide which attacks the intended enzyme and the plant is unable to survive and is thus susceptible. In the next scenario, we have a mutation which results mutation in the enzyme such that the herbicide can no longer bind to the enzyme and the plant thus survives. This is commonly known as the target site resistance. In the next scenario, we have a mutation that does not occur in the enzyme but elsewhere in the genome, such that the herbicide is no longer available to bind to the enzyme. So, for example, let's say we have an enzyme in the cell that would degrade the herbicide, such that the herbicide can no longer bind or attack the enzyme and the plant thus survives and is resistant. This is commonly known as non-target site resistance, and from here on, this is referred to as NTSR. So the NTSR mechanism is commonly a complex polygenic mechanism, but most studies in the literature have identified only a couple of genes that are involved in NTSR, and as such, we are lacking a holistic picture. Multiple questions remain unanswered regarding NTSR, so I begin by asking three questions. What are the loci associated with NTSR? How are these loci maintained over generations? And also, what are the loci associated with the cost of NTSR? So to answer these questions, we chose Ipomia purpurea or morning glory as our study system, which is a common agricultural weed in the US. It is ideal to study NTSR herbicide resistance because multiple populations of the species have been exposed to the herbicide glyphosate, and different populations have developed varying levels of resistance against glyphosate. Additionally, it has been established that this species has polygenic NTSR mechanism against glyphosate. So coming to the first question, which is what are the genes associated with glyphosate resistance? So to answer this question, we resequenced the genomes of a total of 69 individuals, of which 30 were resistant and 39 were susceptible, from different populations shown here in red and blue dots. Red represents resistant population and blue represents susceptible populations. Now, using the variants that we obtained from these 69 individuals, I then applied a two-led approach to identify genes that are under selection for glyphosate resistance. Briefly, as the first step, since I had multiple populations which might compound the results leading to false positives, I first estimated population effect and then identified any outlier loci showing high allele frequency differentiation between resistant and susceptible populations. So for this, I used a statistic called Bayes factor from BANF2 program. On the other hand, I identified regions showing extreme differentiation between the resistant and susceptible populations, which is represented by the statistic GST. Now, to narrow down on the candidate genes, I then performed multiple selection tests like PI, the GMAS-D, Fay and Booz H, and H12, and then combined all of these into one statistic called mid-rank P, which I then used to identify regions under selection. Furthermore, results from both of these approaches were combined to find common genes, which were then defined as the genes associated with glyphosate resistance. So, looking more closely into the results, using the first approach, I found about 1900 genes, which are represented as the red dots. Using the second approach, or the mid-rank P approach, I found about 358 genes, which are represented in the green dots. Now, between these two approaches, there were 128 genes that were common, and the regions carrying these genes are represented as blue arrows here. Now, these 128 genes were then used to understand the mechanistic basis of glyphosate resistance. Now, looking more closely into what genes are present within each of these selected regions. The first region I'll talk about is this region on chromosome 10. This is a highly differentiated region between the resistant and the susceptible population and was under selection for glyphosate resistance using both base factor and mid-rank P. There are also multiple interesting genes that are present within this region. For example, I found 8 copies of glycosyltransferases, which are represented by the khaki lines here, and 9 copies of cytochrome P450s, which are represented by the olive green lines. Both of these gene families have been indicated in the literature to potentially be involved in herbicide detoxification. Further, I found multiple non-synonymous mutations within these genes that are almost fixed in the resistant populations. And as an example, I will zoom on in this region of the 7 copies of glycosyltransferases. So here we have a haplotype map of the tandem block of 7 glycosyltransferases. The gray bar here on top represents exons, stars represents non-synonymous substitutions. The yellow represents a homozygosity state 0 by 0, blue is another homozygosity state 1 by 1, and the pink represents heterozygosity state 0 by 1 or 1 by 0. So we clearly see that the resistant individuals exhibit a stretch of homozygosity as compared to the susceptible individuals. And also that the multiple non-synonymous substitutions seem to be almost fixed in the resistant population. Among other regions, there was this region on chromosome 11, which again contained a copy of cytochrome P450, a copy of glycosyltransferase, and multiple copies of phosphate transporters. Phosphate transporters are important because they have been identified as the transporters that are responsible for bringing in the glyphosate into the cell. So all of this again indicate the role of detoxification. Not shown here, but our gene expression analysis and functional assay also supports the role of detoxification underlying glyphosate resistance in this species. Additionally, within this region of chromosome 11, I also found genes like NAC92, LBD91, and LRK10L, which are all involved in stress signaling and response. Another very interesting region was this region on chromosome 6, which is a part of a longer, highly differentiated region. And this region contained copies of ERF4 and kinases, which have been shown to be involved in stress signaling and response again. I will expand on this particular region of chromosome 6 in a few slides. So we have answered the first question, and it looks like detoxification and stress response genes are the genes that are probably contributing to NTSR. The next question is, how are these genes maintained over generations? So given that we identified multiple genes associated with glyphosate resistance across different chromosomes, next I asked, what force could be maintaining these loci together? Specifically, I asked whether the identified loci exhibit long distance or interchromosomal linkage disequilibrium. So to examine this, I took 4 most extreme regions under selection. Shown here is the linkage value spanning the regions under selection. Here, the lighter color or the more yellow values represent low linkage as compared to the darker or the more purple values, which represents higher linkage or tighter linkage. Now, looking at the regions under selection, which are represented here by the black boxes, we see that these indeed exhibit higher interchromosomal linkage disequilibrium as compared to the nearly zero background genome-wide linkage. So this indicates the presence of inter-chromosomal linkage disequilibrium, which most likely influences resistance alleles found on separate chromosomes and thus potentially mediates resistance through generations. Now coming to the third question, which is what are the genes associated with the cost of NTSR? So one of the important things that mediates the evolution of resistance is the cost that is associated with resistance. And in morning glory, there is a fitness cost that is associated with resistance, such that the level of resistance is correlated with the proportion of germination. So the higher or more resistant individuals show lower germination and deteriorated seed quality as compared to the less resistant individuals. Further, given that I identified regions under selection for glyphosate resistance, I wanted to look more closely into all of these genes in all of these regions to see whether any of these genes could potentially explain or could potentially show any correlation with the cost of resistance that we see in this species. And now I will come back to the region on chromosome 6. So this is a very interesting region for multiple reasons. First, this region exhibits the most extreme population differentiation when comparing the resistant and susceptible individuals. Interestingly enough, this region was also under selection in the resistant and the susceptible populations, but for the opposite allele. So if we look here at the allele frequency map, we see that in the resistant population, if the resistant population carries allele 1, the susceptible populations carries the alternate allele in this location. And although the values are not stated here, but we can see that these alleles are almost close to fixation in the respective populations. Now within these regions, we found two very interesting genes, specifically NFD6 and NACNOT25, and both of these genes are required for normal seed development and growth. Not shown here, but I also found that NFD6 is strongly downregulated in the resistant populations as compared to the susceptible populations. So this indicates that this region might play a role in contributing to the lower germination cost observed earlier. Additionally, we also identified that these genes are very closely linked to the resistant alleles we have identified in this region, which potentially indicates the role of linkage disequilibrium in maintaining the cost in this species. So to summarize the finding, we found the role of herbicide detoxification underlying glyphosate resistance. Additionally, we've showed the potential role of interchromosomal linkage disequilibrium in maintaining resistance alleles over generations. And lastly, we've identified potential fitness cost loci that are strongly linked to the resistance alleles, indicating the role of linkage disequilibrium in maintaining cost in this species. With that, I'd like to thank all the people involved in this project and beyond, and I can take any questions that we may have.